Hi, my name is HChandy. I am a senior at Dulles High School, and my project is over animal models of depression. So before I start, I want to give a quick introduction on depression itself. So major depressive disorder is an affective disorder, which means that it results in impactful changes in a patient's affect, which is their outward projection of their internal mood and state of mind. So depression is characterized by one or more recurring depressive episodes, which consist of at least one of two core symptoms: depressed mood and anhedonia, which is a reduced state of pleasure of normally pleasurable things like sugar or sex. So at least one of these core symptoms are accompanied by at least four additional symptoms, which have been listed below. And in terms of treatment, antidepressants and psychotherapy are sort of the main forms of treatment. However, there are other treatment modalities like exercise, mindfulness, brain scans, things like that that have been explored. But this really depends on what the patient wants and what the patient feels works for them. So it really depends. So animal models, as the name suggests, are animals that are used as models for disorders or diseases, whether common or uncommon. And really the main purpose of animal models is to have to have a means to really explore the underlying concepts of a disease in a way that you cannot research in humans. And so animal models have many benefits, but the main benefit comes in the control. Everything from start to finish is in the control of the researcher. Whereas in human models, that isn't the case. An example of that is with etiology or the cause of a disorder. So in animal models, etiology can be manipulated. You can control how this disease is induced in this model. Whereas in humans, when you get a human patient that you study on like a case by case basis, you cannot control how that human has gotten the disease. The human has already gotten the disease. Another thing is that researchers can limit exogenous variables, such as environment or genetic composition, things like that. And those are things that cannot be done in human models. However, there are complications in animal models, especially when looking at it in the scope of depression itself. One thing is that MDD cannot be fully represented, and that's because depression has human symptoms such as suicidal thoughts, and these things cannot be quantified or represented in an animal model. And another thing is that there are controversies from extrapolating mood from locomotor activity. This is because there's no set relationship between depressed mood and increased or decreased activity, and it's controversial to assume so. So one thing to keep in mind is this overarching theme that a model of depression combines a environmental or stress manipulation with a behavioral assay, which basically examines the effect of this manipulation on the animal. So we're going to look into the latter part of the equation first. So behavioral assay is just a test that examines the behavioral effect of something. And so we're going to be exploring the most common behavioral assays for depression. So two of the most important and most used behavioral assays of depression are the Foursome Test and the tail suspension test, which attempt to quantitatively measure worthlessness, despair, and mood. In the Foursome Test, the mice are placed into these inescapable bodies of water, and in the tail suspension test, the mice are hung on their tails and basically elevated and are not able to escape. So in the Foursome Test and in the tail suspension test, immobility time is measured. And in both of these tests, it's different things. In the Foursome Test, which is pictured below, that's when the mice is simply floating. It's no longer thrashing around or attempting to fight. And in the tail suspension test, which is pictured above, the mice is no longer flinging around. It's completely still. And so when this immobility time is measured, it's used to deem the presence of depression-like phenotypes. Basically, the relationship is that an increased immobility time means that the mice has given up. It no longer wants to fight. And that lack of time of fight is correlated to the presence of depression-like phenotypes, especially when compared to normal mice, because that means that there's higher levels of wordlessness, depressed mood, and despair. So another behavioral assay is the Sucrose preference test, which basically measures the animal model's level of anhedonia, which again is a loss of interest in normally pleasurable things. The mice can either take the normal water or the sucrose water, which is just sucrose dissolved in water, and the spouts are completely identical. And in normal mice, the mice will have a higher preference for sucrose water because it's sweet and they gain pleasure from that. But if there's a reduced sucrose preference or even no preference at all, mice are said to display anhedonia, which can indicate a presence of depression-like phenotypes. So another behavioral assay of depression is the open field test. This measures anxiety-like behavior and locomotor activity. Basically, mice are placed into this box. And mice that spend time on the outer edge, as pictured in the image, mice that spend more time in the outer edge are said to have higher levels of anxiety and lower levels of locomotor activity. Whereas if it's the opposite and they spend more time in the center, they're said to have higher levels of exploratory behavior and lower levels of anxiety. And basically, the normal mice will spend more time in the center because they want to explore. And basically, this open field test is able to see the levels of anxiety and just overall movement as well, and a decreased or increased locomotor activity and higher levels of anxiety can indicate the presence of depression-like phenotypes. So alongside these behavioral assays, there are also other measurements of depression that are used alongside and simultaneously. One thing is sleep, which can be measured by EEG and EMGs, and that's pictured on the right. Basically, the electrical impulses in the mice's brain are assessed and examined very similarly to how these tests are done on humans. And this can indicate hypersomnia or insomnia, which is a symptom of depression. So another symptom of depression is increased or decreased appetite. And by measuring body weight and food intake, the researchers can also see if those symptoms are present. Another measurement that can be done are tasks of memory and attention. And so there are many tests and many tasks that are done within this category, but one of the main ones is the novel object recognition test, which is pictured below. Basically, during training, the mice are presented with two identical objects. And on the testing date, one of these objects is swapped out for a novel object or an unfamiliar object. With normal mice, the mice will go to the novel object, A, because they have higher levels of exploratory behavior, but also because they can recognize that the object one they have seen before and they want to see the unseen object. So when applying this task, researchers are able to see the overall levels of exploratory behavior, but also assess the memory and attention of the mice. And mice with lower levels of memory and attention and lower levels of exploratory behavior are said to display possible phenotypes of depression. And the purpose of these other measurements is to have more evidence and more weight to the conclusions drawn by the behavioral assays. It's just more data to support the conclusions. So now we're going to go on to models of depression. So one thing to note is the field of depression is very vast and very large. And so there's lots of experiments being done all the time in this field. And so I've chosen three models of depression, which I feel are the most relevant to the field today. And even within these models themselves, there are many, many types of experiments done. And so I've chosen one study for each model of depression that I feel accurately sums up the bulk and majority of the research being done for that specific model. So the first model of depression that I wanted to talk about was the changes in diet model. Basically, as the name suggests, their diets are changed in order to induce depression. And the most common way that researchers do this is by giving half of the mice a normal diet and half of the mice a high-fat diet. And a really good example of this is a study conducted by Wu and their colleagues, which basically compared the behavioral and neuroimmunological changes in adolescent male mice. Basically, a high-fat diet is just a diet that has increased levels of fat, cholesterol, and caloric intake. And as you can see from the diagram on the bottom, the mice in this particular study were given the high-fat diet for 6 weeks, and the other half of the mice were given the normal diet. And behavioral assays on both types of mice, both of the groups of mice, were done on the 3-week mark, the 4-week mark, and the 6-week mark. So from the graphs from the data that was collected in the study, we can see a lot of things. The first is that body weight, the rate at which the body weight increased, was significantly higher in the high-fat diet mice. This means that these mice had an increased rate of weight gain. And with this, we can see that there was an increased level of appetite, which is a symptom of depression. Another thing that we can see from the results is that in the tail suspension test and the Foursome Test, which is graphs D and E, we can see that there's higher levels of immobility time. And as I mentioned before, that means that there were higher levels of worthlessness and despair in these mice because they had more time that they were basically giving up the fight. Results of the open field test, we can see that the number of crossings in the 3-week mark was significantly higher, and that in the 4th and 6th weeks, especially in the 6 weeks, they were significantly lower. And with this, we can tell that the mice displayed lower levels of exploratory behavior and lower levels of locomotor activity, which again are symptoms of depression. So what we can conclude from this study then is that the high-fat diet is effective in inducing depression. But one thing to note is that it's hard to determine whether these changes in behavior were done through obesity or because of the depression itself. Obesity is induced alongside depression because of the higher caloric intake. And so it's hard to see what really caused these changes. But a possible solution to this is that this study be done again, or similar studies be done again, but with mice that cannot gain weight. So then that obesity factor is eliminated, because then the mice cannot gain obesity because they cannot gain weight. And so that can be a possible solution to that problem. So the next model that I wanted to explore is the unpredictable chronic mild stress model. Basically, these mice go through very unpredictable stressors. It really depends on the lab, and these are done randomly for many hours each day, and the most common ones have been listed below. And the main underlying concept of this is that these stressors will cause anhedonia and other symptoms of depression, like depressed mood, because physical stress in this model is said to cause depression. But there's a problem with this model because in humans and in real life, not all humans that go through physical stress will get depression. There's something else underlying that makes these particular patients of depression susceptible to depression, even if it was caused by or elevated by stress. There's still an underlying something there that is the reason why only particular people get depression. And the problem with this model is that it assumes the opposite. It assumes that all people and all mice that go through stress will gain depression. So a good example of the usage of the UCMS model is a study done by Wu and their colleagues, which explored the effects of chronic stress on depression-like behaviors. And on the diagram below, we can see that the CMS, the behavioral stressors were done for a period of 6 weeks, and then behavioral tests were done right after. And so it's important to note that from these results that there are firstly 2 strains of mice that were used. And this is important to note because it makes reading the graphs a little easier. So we can see that the first 2 bars are the first strain of mice, and the last two bars are, the last two bars are the other strain of mice. And the CUMS are the ones that went through stress, and the ones that only have a C are the control. That means they didn't go through stress. So from these results, we can see that A, there's a significant decrease of sucrose consumption in the mice, which means that they have anhedonia. They're not receiving the pleasure from the sucrose water. The second thing to note is that there's significantly higher levels of immobility time, which means that they're spending more time dejected and having feelings of worthlessness. Another thing to note is that in the CMS mice, they're spending less time in the center square and also crossing at a less time, which again shows that there's less locomotor activity and that there's a significantly less levels of exploratory behavior and significantly high levels of anxiety. And all of these things together show that the CUMS model, the chronic unpredictable mild stress model, that this is effective in inducing depression. So the last model of depression that I wanted to talk about is the social defeat model. So this relies on agnostic social encounters, which are basically those of aggression. So as you can see from this video, a smaller submissive mice is placed in the cage of a much older and aggressive and dominant mouse. So what happens is that this aggressive mouse chases and basically asserts its dominance over the submissive mice. And not only is this a physical stressor, because again, the mice is getting attacked, but it is also a mental stressor. Because once the allotted time that the researcher determines that these two mice will interact, once that time is over, the smaller mice is placed in a cage within the bigger mouse's cage. And you can see that in the image below. But because of this, even though the bigger mice cannot attack the smaller mice through this cage, a mental stressor is employed because the smaller mouse is afraid, and these feelings of fear and worthlessness and despair, these feelings are still induced even though there's no physical encounter. So a really good study that employs this model is a study done by Borsch and their colleagues, which basically computationally assess the multidimensional behavior alterations after chronic defeat stress. So the thing to note from this is that it was done all computer generated. And from the computer generated results, they were able to conclude that yes, the social defeat stress model was able to induce depression. The more important finding, however, is that the social defeat stress, unlike the previous unpredictable chronic mild stress model that I mentioned, unlike that model, it is able to account for the variability in mice. As I said before, not all humans who go through stress gain depression, and this model accounts for that. We can see that with the susceptible and the resilient mice. The susceptible mice, which is the red, those mice gain depression. And the resilient blue mice, they did not gain depression. Both the susceptible and the resilient mice, they went through the stress. However, only the susceptible mice only in those mice was depression induced, which you can also see in this Venn diagram, which shows that there's a very high similarity levels between the control mice and the resilient mice, which means that the resilient mice did not gain depression. And so that's the most important thing to note from this model, is that it is able to account for the variability and therefore is most likely the most similar model of depression there is with humans. So now I just want to quickly summarize and wrap up and conclude this review showcase. So in the field of depression research, there are many limitations. A few of these are firstly, the fact that there is human presence. When the mice have to go through behavioral assays, like for example, the Foursome Test, the researchers have to physically take the mice out of its cage that it's spending its day in. It has to take it out and place it in whatever testing area there is for these behavioral assays. And even though that human presence may be very small, it still can have a significant impact in the results. The mice may be afraid of this new contact that it's having. It may be anxious because it doesn't understand who this person is or what's going on. And these things can really impact the results of a behavior assay and therefore shift the conclusions drawn from the study. Another thing that is important to note that is a limitation is the overlap between anxiety and depression. So anxiety and depression have many similar symptoms, and a lot of research that has been done to distinguish both anxiety and depression, it shows that there are many comorbidities between the two diseases. And because of this, it's very hard to understand whether the behavioral changes that are observed from the study, whether that's because of the underlying depression or because of the animal's fight or flight response, which is basically what causes anxiety. Thirdly, the problem and a limitation in depression-like research is isolating naturally social animals. The problem is that mice are naturally social animals, and many researchers isolate them so that other mice don't interact with the other mice and tamper results. But the problem with this is that the isolation can cause changes in mood and changes in behavior, which may be mistaken for depression-like phenotypes when it isn't. Possible solutions of this is firstly, human-free systems that are able to study these things without having to rely on human presence, so things that are only being done in the cage. Another thing is researching biomarkers, which is basically researching something that can identify the presence of depression. And if biomarkers have been identified, then there isn't as much confusion as to whether anxiety or depression is being induced. In terms of where I fit, the plan for my future that I want to pursue is that I want to become a psychiatrist or a neurologist. And this would require me to go to med school and specialization. And once that I have successfully gotten into this career, I want to research the subtypes of depression. Basically, depression, based on the path of symptoms it takes, can be divided into subtypes. For example, there are 9 possible symptoms of depression, and there are only at least 5 need to be present in order to get an official diagnosis of depression. And based on which symptoms are present, these depression can be divided into categories. But however, there's not much research being done that solidifies the presence of these categories. And because of this, antidepressants and other drugs that are used to cure or ameliorate depression-like phenotypes, they are only for the main type of depression. They view depression as an umbrella term. This can be viewed in the statistics as well, as 20 to 30% of patients don't gain proper treatment from antidepressants. And so what I would do is research these subtypes of depression and further understand the categories and the multiple variants of treatments that can be taken from there. Lastly, I just want to thank Ms. Bryant for allowing me the opportunity to be placed into this mentorship and also teaching so many practical skills throughout the entire mentorship process. I also wanted to thank Dr. Krishnan for taking me in as a mentee and for teaching me so many field-related skills of research and depression-like research that I can use both in college and in the workforce. But yeah, thank you so much for listening to my presentation. And if you have any questions, please feel free to ask. Other than that, you are free to join another one of my peers' presentations by going through breakout rooms, and if there's no questions, then I will be starting over the presentation. Thank you again.